Hello again, fellow humans, and welcome to another biology video. This one, as you can see, is about mutations. And this is where we're going to start getting into some of the practical applications of what we've been learning so far about protein synthesis. We know that the DNA has this information on how to build proteins, and the protein leads to our traits. And so, what we want to know is what happens when you change the DNA and how is that going to affect the proteins and our traits. But before we get into that, I want to start with a very simple question. I ask this question pretty much every year. You can see it at the screen, at the bottom of the screen. What do you think of when you hear the word mutant? Now, of course, I don't know what you just wrote in here in the ed puzzle, so maybe what I'm about to say has nothing to do with what you were thinking. But overwhelmingly, in my almost 10 years of doing this now, the answers that students give have to do with Ninja Turtles and X-Men, right? And I'm sure a bunch of you said Ninja Turtles and X-Men. And so that's interesting. Like, why are these cartoons what we think of instead of like the science? But that's, of course, probably because people don't sit around talking about mutations and genetic diseases all the time. And maybe you do enjoy reading comics or watching movies or cartoons that have to do with these mutants. But it's unfortunate that that happens because it gives people the impression that mutants and mutations are always bad. I mean, literally, these are monsters. If you were to encounter a 6-foot tall talking turtle that came out of the sewer with ninja weapons, you would not be going up to them just like trying to be their friend, right? Like you would be running away screaming because that would be weird. Of course, in the X-Men, the X-Men are mutants and treated horribly by the regular people because they're thought of as monsters. But we need to get away from this idea because mutants in biology don't mean monsters. Mutants in biology means one thing, that there has been a change in a DNA sequence, right? In this particular case, there's a C right there that got changed to a T. And what was supposed to be a GC in the original DNA is now an A and a T. And like, what happens when you do that? Like, that's what we want to know. But it's important to know that these things are always happening. They happen whenever we're copying DNA. And so there's a step where you copy DNA called replication that we actually haven't talked about. But this is where you make your body cells and then something called mitosis, or your sex cells, your sperm or your egg cells in something called meiosis, right? And so you're constantly making new cells, you're constantly making your sex cells. And as you do that, mistakes are going to be made. And in fact, if this is the definition of what mutation means, that means that everybody is a mutant because everyone has little mistakes that have been made. And to take that even further, those mistakes are how all the differences in all the different species and life on Earth, this is where all the differences arise from. All the differences between humans and other species is because of mutations. And especially if you don't have dark hair, dark eyes, or dark skin, then you are absolutely a mutant because our ancestors all started with dark hair, dark eyes, and dark skin. And anything other than that is the result of a mutation. Now, of course, mutations are going to lead to a number of genetic diseases, but they're not always bad, as we're going to see. One little thing is that technically mistakes could happen during transcription when we're taking our DNA and turning it into RNA, but we don't care about that because the RNA is only temporary. And so if a mistake was made, it would be bad for a minute or so, but then you would recopy your DNA into new messenger RNA, and then it probably wouldn't make a mistake and everything would be okay. So we don't really care about that. We do care about mutations in our DNA, though, because those are permanent, at least permanent in every cell after the mutation. So if a mutation happens, say in, where am I going to put my head this time? Down here is fine. If a mutation were to happen in one of your sex cells, called a germ line, this would mean that if that sex cell was used to make a baby, then that baby would have that mutation in every one of its cells because the first cell had a mutation in it. Um, if that mutation happens in what's called a somatic cell, it's a body cell, right? If it were to happen in your shoulder, that would only affect any cells that came from that part. And so maybe in that little area of your arm or shoulder in this case, there would be something weird, but it wouldn't be everywhere in you, and it wouldn't probably cause that many problems. But the mutations that you inherit from your parents, those are the ones that are going to lead to the differences in appearances or traits or whatever it might be. So let's get into some examples, right? The best way to do this is to just look and see how it affects the protein synthesis. So I have my original DNA here, as you can see, and a mutant sequence. And in that mutant sequence, I have changed a G to a T, as you can see, right? So we take our sequences, and there they are, side by side. Of course, the mutation is right there, and it's going to be carried over into the messenger RNA. During transcription, what should have been a C is now going to be an A. Now, what does that mean? Well, remember, we have to look at codons. So real quick, there's our first codon, second codon, third codon is GCA. But in this one, it's GAA, right? So we go to our codon chart, GCA versus GAA. And GCA makes something called alanine. And GAA makes something called glutamic acid. Changing the one letter has changed something about the codon, which has led to one amino acid being different, right? The first two are fine because nothing changed there. The last one is fine because nothing changed there. In the specific spot where we have substituted a letter, right? I substituted a T instead of a G. This has led to one amino acid being changed. Now, hopefully you remember that the shape of a protein is dependent upon the order of amino acids. And so if you change the order of amino acids, you might change the shape. You might not. It's a little bit tricky in this case because it's only one thing, right? Like it's just one out of, in this case, 1 out of 4. But in reality, it would be 1 out of hundreds, if not thousands of amino acids. So in a lot of times, this might not make any difference. And you probably have mutations like this in your DNA, but they just don't matter that much. But they can matter a lot. There is a genetic disease called epidermolysis bullosa. This is a disease that has to do with a protein called collagen. Collagen is supposed to be a long, stringy protein, and it's kind of elastic, and it's all over your body, but it's definitely found in your skin, and it's what lets your skin be flexible, right? Like if I have, hopefully you can see that, right? When I pull up on my skin, it just snaps back into place because there's these elastic proteins that are long and stretchy, and they can keep my skin in the shape that it needs to be in. But changing just one letter, just the wrong letter, it changes just one amino acid to cause the collagen to break into little pieces, and it ends up being very fragile, and your skin is kind of like dry and papery. I'm going to do this real fast, but it leads to basically your skin kind of falling off. So there you go for a really freaky picture of having skin with a genetic disease caused by one mutation in your DNA or this person's DNA. That's enough of that. Right? So this is called a point mutation. There are 3 basic types of these point mutations. All of them happen when you change one letter. Right? It's also called a substitution. The one that we just talked about is called a missense because it kind of makes sense, but there's a mistake in the chain. Right? Again, this could lead to a lot of changes in the function, but it might not make any difference. We kind of just have to see what happens. The technology and the science actually hasn't gotten good enough yet that we could predict what's going to happen. We kind of just have to wait and see. And if somebody gets born with a mutation and it ends up being bad, then we know that it was bad. We don't really hear about the ones that don't make any difference because, of course, it doesn't really attract any attention. Now, there is a way to definitely mess things up, and that would be something called a nonsense mutation. It's shown down here, and that's where an accidental stop is put in, right? We changed one letter, and it makes a stop in the middle where it shouldn't be, right? Stop should be at the end. So this is clearly going to be bad if you're trying to make a particular product and you stop partway through, then you're not going to get the right shape that you're supposed to get. Now, there is a third version of this to talk about. Let's say this has happened. I missed my title slide here, but somewhere in here is a change. Did I put it there? There it is. There should have been a T, and instead there's a G, right? One letter has been substituted for another. This is going to carry over into the messenger RNA. And again, here's our codons. So now it's still GCA, but this time, instead of GCA, it is GCC. And so we go back to the codon chart. GCA still makes alanine like we knew before, but GCC also makes alanine, right? And that's actually pretty awesome. This, as you're going to see in a second, is called a silent mutation. You might have noticed that there are 4 different ways to make some of these. Some of them have 3 different ways. A few of them have 2 different ways. Some of them actually have 6 different ways. I forget exactly which one it is. It's arginine right here. Arginine has 4 here, and there's 2 more over there, right? And so this is actually a cool way to avoid bad mutations because the change didn't actually change anything. The DNA is different. It is a mutation, but the final product is completely unchanged. And so, again, this is, even though you undoubtedly have lots of mutations in your DNA, hopefully most of them are like this. They are silent mutations that just don't happen to change anything. Right? And so here again is just in words. There it is. And again, you can kind of see the 3 different pictures. A stop, which would be the worst. A missense, which changes one amino acid from what it was supposed to be. And then a silent mutation, which doesn't change anything. Right? So those are called substitution mutations. Now, there's another way we could mutate things, and that is to delete or add nucleotides. Believe it or not, this happens relatively frequently. In this case, I have made a deletion. I have cut out that C. The 3rd letter has been deleted. You can see that everything else afterwards is the same. It's just the C's gone. So just like before, we don't really know what's going to happen until we play it out. Here's the original versus the mutated sequence. Again, the C right there is gone. It should be right in that spot, but it's not. And that leads to a G that should have been in the RNA from being missing, right? Like it's just not there. And so then we carry it over into our codons. And you'll notice that the first codon has changed, right? UGG is now UUC. Sorry, I said UGG. It's UUG versus UUC. But that's only the beginning of the problems because the next codon was supposed to be CUA, and now it's UAG. Completely different. The 3rd codon, GCA, is now CCG. The 4th codon gets cut off in this particular sense. Remember that there's actually more letters over here. I'm just not writing them. So technically, there is a 4th codon right here. It's just I have to stop writing the letters at some point. So now we go and we use our codon chart and we get our amino acid sequence. And the first one is different because of the missense, right? Just one letter was changed, lysine to phenylalanine. But then the 3rd codon got a stop, so I made a nonsense on accident. The 4th codon was supposed to be alanine, and now it's proline. And so, maybe you can see that this pattern is going to continue down the line. The next one is probably not going to be valine. It's going to be something else. Because a deletion is going to shift everything around and make all of the amino acids change after the point of the mutation. This is a serious problem, right? Now, we're not just changing one thing, we're changing lots of things. The same thing would happen with an insertion. In an insertion right here, I have inserted a C that wasn't right there, right? Like I've just put it in there. And so again, The extra C is right here. That gets carried over to an extra G right there. We put our codons. UUG is the same. Notice this happens before the mutation, so nothing is going to change there. That one escaped. The second codon has one letter that's different. It's a CUG instead of CUA. But then again, here's where the major changes start to happen. The third codon is totally different. The fourth codon is totally different. All the codons are going to be totally different. Now, in this case, when I went to the amino acids, the lysine the same because that's the part that never got changed, right? That was before the mutation. The second one was changed, but I ended up making a silent mutation right there, right? That's just randomness right there. I didn't plan that at all. But then the last two are totally different, and you would expect all of the ones that follow that I didn't put, they're all going to be different. Because once again, you have moved things. And that's the key to this whole thing, right? Because we have to read them in groups of 3, you can kind of put these boxes, like remember I had you practice that once. I don't know how many people still do that. But it does help to visualize that when you say insert something, it pushes the letters into a new box, right? Or if you delete one, it pulls the letters into new boxes. And so the boxes are sometimes called frames. And so this is called a frame shift mutation. It's going to change the whole sequence and lead to a lot of differences. Oh, that one changed too. I forgot about that. So here's the kind of summary of the whole thing. Frame shift means that we're shifting the letters because the codons are being moved. Insertions or deletions are going to lead to the same issues. There is an interesting one down here in this diagram where they cut out 3, right? And this could happen, actually. You could cut out sometimes entire chunks of your DNA get deleted. That's obviously not a good thing. And deleting 3 isn't a good thing. But actually, deleting 3 is better than deleting 1 or 2, because deleting 1 or 2 leads to a frame shift. Meanwhile, deleting 3 just completely erases that one amino acid, but all the other ones would stay in their frames. Notice these here are the same as they were supposed to be before, right? And so that's a little quirk about things. I'm not going to mess with you in that case, though. You can be certain that most of the ways that you're going to see these happen on worksheets or whatnot, it's going to be a deletion of 1 or 2. Now, mistakes happen a lot, right? When you're copying your DNA, the enzymes that copy things go real fast, 1,000 bases per second. So they make a lot of mistakes. Maybe you don't think 1 in 10,000 is a lot. And I guess it's not really a lot, right? I imagine if you did 10,000 math problems and you only got 1 wrong, like you'd be doing pretty good. But your DNA has 6.4 billion letters in it. Is that a billion? Pretty sure that's a billion, right? So if you make 1 mistake every 10,000, you're still going to make 640,000 mistakes. That is a lot of mutations to have inside of your DNA, right? Not good. But thankfully, there is another enzyme that you don't need to know the name of it, but you should be resting easy that it can go through and fix problems. This image over on the right is trying to show that there was a mistake made right there, right? And of course, these are all shapes. So if you make mistakes, what you really end up with is a weird shape that happens. You can cut that part out and then rewrite it, do it over again, and fix it. And so in this case, it's going to take the error rate from 1 in 10,000 to 1 in 100 million. So now our 640,000 mistakes becomes 64 mistakes. That's much better. You could still expect 64 mistakes to be made every time you make a new cell. And again, this is happening all the time, right? Especially in your skin or in your hair cells, your hair follicles, where the hair comes from. Your hair itself is not alive, but that's a whole other story. But in any case, the only ones that you really care about are the ones that are going to be in your eggs or your sperm cells. Because again, like say if I get a mutation in my arm right here that messes up my collagen, I might have a little spot on my arm that ends up with that mutation and ends up having the collagen that's weak and fragile, like in that picture. But that's just that one spot, right? Um, so again, for you, your body's mostly grown, so you don't really need to worry about mutations in that sense. We do need to worry about mutations when we talk about cancer, but we are not ready to talk about that one at all. That comes way later. So, in any case, we heard about one of a genetic disease called epidermolysis bullosa that's related to a mutation. And as we go forward through this unit into next semester, we will stop many times to talk about genes, the proteins that they make, and the jobs that those proteins do. And then talk about what happens when you break them, when you get a mutation and the protein doesn't work for how it's supposed to. But again, that is, we're working our way up to that.